Thanks very much, Jake. Okay, um, so I'm going to talk about something I'm very excited about. I think a lot of you are very excited about biological networks, and I'm particularly talking about challenges in representing biological networks, from nodes to edges to interfaces. So Jake actually gave a very nice introduction to biological networks, and this worldview of thinking about not just nodes, but also edges connecting those nodes. I mean, it's very, of course, these days everybody is very familiar about this, but I think it's fair to say that when this worldview first came about 20 years ago in the late 1990s, it was a truly revolutionary. You can have this nodes and edges worldview, and as a unifying language, right, to talk about a useful language to talk about diverse scientific disciplines ranging from social science to computer science all the way to life science and medicine. So of course, this is also how a lot of people are thinking about these days about in the field of systems biology, right? So when you think about systems biology, we think about people doing large-scale omics analysis. And then one of the goals of systems biology is to try to build a model, right? Try to build a model, let's say, of a human cell. A model which doesn't have to be extremely detailed, but it needs to be useful. It needs to be useful in the sense that it can give you some useful information by making some useful predictions. But how do you go about building such a model? So one of the popular ways of thinking about how to build a model for the human cell is to view the human cell as a network, right? As a nodes and edges, where each node is let's say here is a protein physical interaction network. So each node is a protein or other kinds of biomolecules, and each edge is an interaction. In this case, a physical interaction between two proteins and other sort of molecules. So of course, there are many other ways of thinking about cellular circuitry. And there are many different types of nodes and many different types of interactions, ranging all the way from physical interactions to functional interactions, all the way to genetic interactions, and many other ways of thinking about these interactions. Now, but is this really the best way of thinking about building a model? This is up for debate, right? Because after all, thinking about human cell as a biological network, it's a very abstract way of thinking about a human cell. But still, one could argue that, networks represents a sort of a good balance between having too much details in the model versus give you some useful information and be able to parameterize it well and give you some useful information prediction and sort of achieve that good balance between the two. Now, I want to talk about several challenges from my personal perspective, and as well as the perspective from where I think the field is trying to go. So I want to first talk about one of the central challenges in network biology. That is, how do you go from a node-centric view to an edge-centric view? And then next, I will talk about trying to do multi-scale modeling of biological networks, and then here is really trying to bring back atomic details of these nodes and edges into this model, into this graph theoretic model of the cell. And then finally, I'm going to talk a little bit about how to think about assigning functions to these interactions, to edges. So I think it's fair to say that even though everybody is really looking very excited and thinking about network and systems biology, I think it's fair to say that much of systems biology is still very much focused on nodes rather than on edges. And you can, we typically think about genes, think about functions of genes, and people do a lot of curations of the function of genes in terms of gene ontology, which is a huge effort in terms of trying to curate the sort of representing the functions of genes in a way that machines and computers can understand. I think Jake sort of alluded to this a little bit. And when people do experiments trying to perturb these complex molecular systems, typically these experiments are done in the realm of perturbation of nodes, right? You do transcriptomics, you up or down regulate gene transcripts, you knock out genes and do gene knockout experiments all the way from yeast to human and try to study the resulting phenotype. And then because of these experiments and because of these annotations, manual annotations in terms of gene ontology, a lot of the analysis that we do are really focused on nodes or sets of nodes. Genes, I think the primary example is gene set enrichment analysis. You get a set of genes, and then you try to understand what are the functions that are enriched or depleted in this set of genes. And then the edges are really just kind of there to help us understand the functions of genes, of nodes. So what I want to argue is that we really need to think about edge-based analysis is very much needed, but it's a very challenging thing to do. And the reason why this is so challenging is because it's very difficult to pinpoint. Let's say you want to do function of edges. Let's say here's a protein-protein interaction, and you want to study its function. It's very difficult to perturb just that protein-protein interaction and be able to study the function of just that edge. It's very difficult to do, but I think that this is something that I think we need to think about. So we need to think about how to think about functions of not just nodes, genes, also functions of edges, protein-protein interactions, other sorts of molecular interactions, and perturbation of these edges, analysis of these edges. Okay, one of the I think one of the key concepts in this notion of moving from a node-based to edge-based worldview is the notion of ageotype, which is proposed by Mark Vidal from the Dana-Farber Cancer Institute. So the basic idea is that you've got all these mutations, many of them cause disease. You want to understand which mutations are causal, causes what disease. So you can have a lot of these different mutations happening on the same gene, on the same protein, but many of them will actually disrupt the function of the protein in precise, different, subtle ways. So here is an example where we have the wild type protein interacting with two other proteins, and then we have 4 different mutations on the same protein, but they will give you different network perturbation patterns, right? So the first mutation doesn't do anything to the interaction network. It lies on the surface of the protein, outside of the interface. So it's a benign mutation. The second mutation kills the whole protein. It lies in the center of the protein, and it's a very bad mutation. The whole protein falls apart. So basically, you're losing the protein as well as you're losing all the interactions mediated by that particular protein. So that is called a quasi-null mutation. The first one is called quasi-wild type because it's similar to wild type. But then you have all these other very subtle mutations. So for example, the third mutation, it doesn't kill the whole protein. Rather, it killed a very specific subset of the function of the protein. In this case, it lies on the interface of one of the interfaces of the protein, and then it kills just those interactions mediated by that particular interface, but otherwise the protein is still largely functional. So this is a very precise and very subtle way of losing the function of the protein, and this is called agetic mutation because this mutation causes an edge to be lost, an interaction edge to be lost. And then finally, you may have a mutation which actually causes a gain of interaction, an edge gain event. So many things can happen, and these different events will give rise to different phenotypes. So the precise way that each one of these mutations is disrupting the interactome is called ageotype. And this is very much similar along the lines of when people think about genotype, the relationship between genotype and phenotype. So that's shown here. And Steve gave a really nice introduction to this, to the importance of this connection between genotype and phenotype. And then we can think about this ageotype as sort of the intermediary link, the missing link that connects the genotype to the phenotype. And the idea is that if you know the ageotype, right, of each one of these mutations, then we can use this information to predict what sort of phenotype, disease phenotype, that these mutations will give rise to. So you can do this, but then the question is, how do you actually measure these ageotypes of these different mutations? Now you can do this experimentally. That's what Mark Vidal and his collaborators did using many different techniques. You can use H2 hybrid screening. You can use many other screen techniques. So this is primarily done by Nidhi Sawhney and Song Yi from Mark's lab, and Jasmine Coulomb Huntington, who did the computational analysis. So, but these experiments are extremely time consuming. You can only do this for a small number of disease mutations. But they are very useful in the end. So this is an example showing you that you have 4 different mutations on the same gene, and then the first 3 mutations give you the same agiotype. They disrupt the interactome in the same way, whereas the last one disrupt the interactome in a different. In fact, the last one doesn't even disrupt the protein interactome. So presumably it has some other actions on some other parts of the human interactome network. Could be DNA or other molecules. But the basic idea is that the first 3 who give you the same agiotype, they will actually in the end give you the same phenotype. They cause the same disease, whereas the last one give you a different disease. So, and the other thing that's also interesting is when you compare disease mutations, Mendelian disease causing mutations with non-disease mutations, common mutations, and you can see that they have very different profile in terms of disrupting the human interactome. If you're non-disease variants, vast majority of them don't do anything to the interactome. They're all pretty much all quasi wild type. But if you got disease mutations, half of them will disrupt the interactome one way or the other. Out of those half, a quarter of them kill the whole protein, so the quasi-null mutations. They kill the whole protein and they kill all the interactions associated with it. But then the other half are those very subtle agenetic cases. They don't kill the whole protein. The protein is still largely intact. They only kill a specific interaction. They only cut a specific interaction that's mediated by that particular protein. So what this tells us is that it is actually very important to carry out these kinds of mutation agiotyping. But the challenge, of course, is that by doing these kinds of experiments, it's very difficult to do. You have to do you have to do these experiments every time you have a new mutation. So is there any a better way of doing these? Well, I can do this using computational methods if you know the 3-dimensional structure of these proteins and protein-protein interactions. So that's something that we're very interested in doing, is to be able to build 3-dimensional structural models for these edges. So this is sort of like a next level representation of biological networks. For some kind of biological networks, in particular physical networks, we can build structural models for these nodes and edges using a variety of different methods. We can do this using tried and true homology modeling methods. We can also do this using more adventurous methods based on alpha folds or other kind of deep learning based methods. And we can use this approach to build these structural models for a variety of different kinds of interactome networks. So in the very beginning, we're interested in disease, in particular infectious disease. So we built structural models for human virus protein-protein interaction networks, which give us a lot of insights into the principles underlying human virus protein-protein interactions. And this work was initiated by Eric Franzosa. And later on, we got interested in using this to look at a genetic disease to understand the impact of mutations in terms of agiotyping, so computational agiotyping, so to speak. And this is the work initiated by Mohammed Ghadi. So it turns out that we can actually, once we have these structural models, we can actually predict the agiotype for these mutations. We don't have actually to do experiments. We can just look at where these mutations are. Are these mutations lying on the surface of the protein? Are they lying on the interface of the protein? Or are they lying at the core of the protein? And then we can use that information to carry out a combination of physics-based predictions and data-driven predictions of the impact of those mutations, the precise impact of those mutations on protein-protein interaction networks. So you can predict, is this mutation going to be quasi-wild type? Is it going to kill the whole protein quasi-null, or is it going to disrupt specific protein-protein interactions? And you can compare our predictions with experimental results done through agiotyping and the results are broadly consistent with each other. So I want to last touch upon thinking about assigning functions to edges, so what I would like to call functional interactomics. So the idea here is if you want to be serious about thinking about edges, sort of having this edge-specific point of view, we have to be thinking about how do you even assign functions to edges, right? Traditional, the way that people do functional genomics, you would knock out a gene, you would knock out genes and then study the function, the resulting phenotype. So that's functional genomics. So can you do a similar thing here? Basically, you knock out an interaction. How would you knock out an interaction? Let's imagine that you pick an interaction, you kill it by some way, and then you ask the question, the most fundamental question you can ask is does the cell feel pain or not? Right? Is this a neutral perturbation or is this a deleterious perturbation? And then the most fundamental question you can ask is what fraction of these edge cutting events are neutral versus deleterious. So you can ask. So that I think this is a very sort of like the first question you can ask. And this is work done by Mohammed Ghadi. So we can, unfortunately, we don't have such an experiment, but we can use sort of computational means to get an answer to this question. Essentially, the way we do this is by just looking at the mutation agiotyping experiment. So I already told you that when you do computational experimental agiotyping, you see that many mutations, disease mutations, disrupt the interactome network, and very few normal mutations, not normal, but common mutations, disrupt the interactome. So what that tells you is that gives you a hint that many of these interactions are actually important. When you kill them, when you kill these interactions, it'll give you a deleterious phenotype. Okay. Now you can make this argument more precise using the Bayes rule. You just need to flip the two sides of the conditional probability, and then you can actually get at a estimate for what fraction of the edges in the human interactome when you cut it, it's actually neutral. It's a small percentage. It's about 20%. So the majority of the human interactome, when you cut each edge, the cell feels pain. I mean, it's a deleterious cut. So I want to provide a summary to this. I think that it's imperative to really think about this edge-centric worldview and generate and develop large-scale, unbiased, accurate data sets and methods in terms of function perturbation analysis of these edges. And secondly, I think the structural system biology approach provides a multi-scale framework where you can actually link the causes, which often happens at the atomic level, and the consequences at the network level are modeled together. I think this is sort of a prerequisite for building a useful model of the cell. So with that, I'll thank you for your attention.